Good afternoon. I'm Dave Miklas here at the DNA Learning Center. We're here in the Laurie Landau studio where I'm bringing you a webinar on the blue line of DNA subway. We have about 12 people online, so that's great. I'm going to show you how to use this tool, give you a little background on why it's important and how you might use it. Before we get started, I'd like to let you know a couple of things. If you click on anywhere in the video, you'll get a prompt for high definition. So if you have the bandwidth, you may want to click that for a better view. Secondly, if you look at the top right on your screen, you'll see a full screen icon. If you click on that, you'll get a broader view. And finally, if you have any questions for me during the webinar, or especially at the end, use the chat function, because that's an easy way to get me a question. So without further ado, let's launch into this. First, I'll give you a little bit of background about why this is important and why you might use it in your teaching. As many of you know, I've been involved in the effort to bring biotechnology into classrooms for some time. And this struck me as an interesting illustration, which is in the 1950s, if you wanted to analyze a single piece of DNA, a gene-sized piece of DNA, it would have taken a centrifuge, ultracentrifuge like you see in this photo here, that would have cost about $500,000 in 1950s dollars. And that centrifugation, just to separate out a gene-sized piece of DNA, would take you probably overnight. In 1973 at Cold Spring Harbor Laboratory, 3 scientists here invented what we now know as agarose gel electrophoresis, which could separate a gene-sized piece of DNA for $250 in about 45 minutes. So the point I want to make here is that as we improve science, one thing we can do is make it more egalitarian, which is to lower the threshold for people to do important experiments. And we've been busy doing that at Cold Spring Harbor for some time. And I'm happy that I've maybe helped some of you or other people do some of these sorts of experiments. But we also now have, if we had an egalitarian gene made possible by a gel electrophoresis chamber, now we have what you might call an egalitarian genome, which has been made possible by new methods for so-called next generation sequencing. As you may know, to sequence the original human genome cost about $3 billion, and it took 13 years. You can now sequence that same genome about overnight for a cost currently of about $2,000 or $3,000 per genome. And sometime soon, we've been promised this sort of plug-in for our computers, which may do your genome for less than $1,000 in a few hours. So now, genomes are available to just about anyone to analyze. And if you doubt me on this, here's a graph about the lowering cost of sequencing 1 million base pairs or a megabase of DNA. And you'll see that for a long time, we followed Moore's law here, which is doubling the capability about every 16 months. And then we really dropped off here beginning in about 2007 with the advent of so-called next generation sequencing. The cost of sequencing 1 million base pairs of DNA has dropped dramatically in the last 5 years, down 10,000-fold. And just try to think of anything in your life that has dropped in price by 10,000-fold in 5 years, or even 2-fold. So we have a glut of data in biology. If you didn't know this, we don't even have internet capability of transferring, say, a big file that has a whole human genome or other genome on it. You have to actually trade these by the mail on USB sticks or other sorts of media. We also have other sources of big data in biology. A big one is from visualization, and another one is from phenotypic data of the sort that you could, say, gather by flying a drone over a cornfield and getting data points on how plants are growing in that field. Well, the availability of all these data has introduced a paradigm shift in biology. I don't think I need to tell you, but gone are the days where we made a hypothesis and tried to cobble together a rare amount of data to support that hypothesis. Now we have lots of data and relatively few hypotheses. So it creates whole new ways of thinking and going in the world. And to my mind, it creates the first time in the history of biology or any science where students and teachers can work together with high-level scientists using the same data at the same time and the same tools. And today we're going to see a demonstration of that, especially using online tools for DNA sequence analysis. And all of us are somewhere here on the continuum of research and education. We sometimes move along this continuum, but to my mind, this middle ground between research and education is really a fertile place for growing new ideas among students and teachers. This webinar is being brought to you by the IPlant Collaborative, so just a couple of words about it. It's an NSF project now in its 6th of 10 years to develop cyber infrastructure for biology research. Although we're called the IPlant Collaborative, we're also branching into animals and other creatures. It's really based on the NSF's vision for how cyber infrastructure should be assembled in the 21st century. And here you have some of the principles of cyber infrastructure. And we're dealing with one of these elements, especially today, which is a virtual organization that allows people to connect and do things in a virtual world. If you're not sure what a cyber infrastructure is, frankly, I'm not either after 5 years, but it involves a lot of hardware, which is called the lowest level of the infrastructure, a lot of systems that allow these hardware to communicate with one another, ways into those data, and finally, into those tools and services, and finally an interface such as we're going to use today that allows a mere mortal such as you or I to access all this high-power computing. And I'll tell you that among the high-power compute capability of the IPlant Collaborative is access to the XSEED supercomputing system of the National Science Foundation, also called high-performance computing. And all of us hopefully can be online with that in just a year or two. IPlant is designed to solve problems in biology, and here's some of the problems that you may have: managing data, integrating different tools. If you're a programmer, accessing the infrastructure at a level called the application programming interface, which really lets you work with the raw tools yourself. Lots of tools and services, but especially for us in biology, this sort of central problem, which is how does genotype relate to phenotype, and also in the presence of the environment. Well, the key element of the IPlant cyber infrastructure is what we call the discovery environment. It's essentially an app store for lots and lots of applications that could help you solve many problems in biology. I encourage you to look at it and to explore it for the tools that may help you. But today, we're going to use the sort of educational analog to the discovery environment, which we call DNA Subway. And here's DNA Subway. It is, of course, a metaphor for a subway or a train line where you have different lines designed to take you different places and stops along the way that you can stop and do different things. We developed this over a period of about 3 years, had a lot of collaborators around the United States and some around the world. Have quite a few users using this, and I hope you may become one. There are 4 lines. The red line, which is used for annotating big pieces of DNA up to 150,000 base pairs. The yellow line, which is for searching for homologous sequences within a sequence genome, especially good for looking at transposons and other repeated sequences. The blue line, which we're going to use today, which is for comparing DNA sequences and making phylogenetic trees. And the green line, which will be out in just a month or two, which will allow you to analyze next generation sequence using high-performance computing. Well, let me tell you a little bit about DNA barcoding, and then we'll launch right into some experiments. I also put on here human mitochondrial DNA sequencing, but I call these sort of do-everything labs. They're simple, they're iconic, they center on the student and projects that they can initiate on their own. They're distributed projects where different students in different ones of your classes or in different classes around the country or the world can work on different parts of a similar project. It integrates genetics with ecology, conservation biology, and evolutionary biology, something that all of us, I think, strive to do. And importantly, it combines in vitro or wet experimentation in biochemistry with in silico or computer bioinformatics on your computer. And as I'll show you, gives the opportunity for novel, relatively simple but novel publications by students especially publication of novel sequences to GenBank. Well, I'll be showing you resources that are available free on the web and in other places. I can tell you that the barcode experiment is available as a kit from Carolina Biological, as well as other suppliers. I have a link there to one of the kits. Although, in fact, if you look at these DNA barcoding links, which you'll also find on the side of your screen, they have protocols for doing these experiments that would let you do these without buying a commercial kit. And all these experiments are also contained in a new book that I put out with some colleagues just last year called Genome Science. So just be aware there's links to resources. I won't be calling on them all during this session, but you can certainly call on them by clicking on them in the resource area to the right of your screen. Well, I said that DNA barcoding provides a pretty low level threshold for having students do a real scientific publication. Here's a brief case study. These were students in New York City who were interested in finding out what kind of sharks are used in the dried shark that is used for the Chinese delicacy shark fin soup. And they found a number of different species could be in those dried sharks. One of them is actually the winter skate here. And here's their work in DNA barcoding, which identified their two samples as a winter skate with some new sequence polymorphisms here and somewhere else. Here's the publication that they did to GenBank. You can see it on GenBank if you do a search for this species. And these are the students and their teachers who were involved with that work. Well, let's talk a little bit about DNA barcoding. This is a universal product code, such as you would find on items that you buy in a store. And this is a DNA barcode. If you've never seen one, these are samples that we collected here at Cold Spring Harbor. And these are samples that were matched from GenBank. And what you'll see, each place where you see a line is a sequence polymorphism or a single nucleotide polymorphism. And what you'll see is that a group of related species, these that we collected here at Cold Spring Harbor, have a related barcode, and other groups of barcodes shake out in other groups of living things. These barcodes are fairly universal. They, in many cases, just these three main barcodes that I show you here can get you down to the genus or species level in many taxa, but in some taxa you'll have to rely on different barcodes. Two main ones used in plants are derived from genes in the chloroplast. Each one of these barcodes is 600 to 700 nucleotides long. And the major barcode used for animal species is in the cytochrome oxidase subunit 1 of the mitochondrion. I don't have it here, but in fungi, another mitochondrial sequence is used called ITS. Just to remind you that these organelle genomes, here's a human mitochondrial genome, are in fact circles. They look like bacterial plasmids because, in essence, that's what they were at one time. Well, let me show you what we would do on the front end of a barcode experiment. That is to develop the DNA sequence that we'll analyze today. First off, everything you need to know is on this online protocol, which you can print out as a PDF. First, you collect samples, and I'm going to show you some samples that I collected on a field trip to Nigeria, but you can go into any sort of store. You can take plants that are growing. It's very easy to do. These are middle school students working in our lab, and these are some high school homeschooled students working in their home kitchen with the cat there. We extract DNA by one of several protocols. Some of them are very similar to a mini prep using an alkaline lysis. Some use a spin column. Those are a little bit more expensive. We're moving towards a very inexpensive one, which we'll publish just momentarily on our website, which uses silica. It's very, very cheap and works as well as a spin column. So it's quite a good advance, making things cheaper and easier. This is the sort of equipment it takes to extract the DNA, then take the extracted DNA and amplify the barcode region from whichever barcode you would like to investigate. And ultimately, after you do all this, and of course it's quite easy and fun for students to do, you end up with a gel that shows you that you've amplified the barcode region. Once you know that you've successfully amplified a barcode, you divide your sample up usually into, the remainder of the sample you divide up into 2 fractions, and you send these out for DNA sequencing. As we'll see in a moment, if you'd like to get data that can be published, you will do a forward and a reverse sequence. If you'd just like to do a classroom activity, a single forward sequence is enough. These are sent off to a company that we partner with called GeneWiz. They clean your DNA, sequence it, and place the sequences on the DNA Subway server within 48 hours. So it's a very rapid turnaround. We have found the sequence to be very, very high quality. Virtually any DNA sequence that generates even a faint band on your electrophoresis gel will give you a decent sequence. And the cost of $3 per forward or reverse read is $3. That's about $1 less than you might normally pay. Okay, here's the experiment that we're going to look at for the rest of the afternoon as a little example of what you could do. In January, I went to Nigeria. I worked in this lab in the eastern part of Nigeria in the city called Enugu. We collected over 40 specimens. We analyzed them, and it was really quite interesting because 50% of the things that we were able to get DNA sequence from were either a species that was not yet in the GenBank database, which is fun and interesting to have that, or had new sequence polymorphisms that hadn't been reported before. And in fact, this is an awful lot of fun for anyone, for me, you, a student, or anyone, to find a species that's not in the database or new polymorphisms that have not been published. And we're actually in the process of publishing these records right now. Well, here's the university in Nigeria that I went to work at. We piled about 20 teachers into this van here, went out over rough roads into the countryside, and started collecting things that we found there. There was a mixture of both planted species, because this had once been the home of a Catholic priest, but there were also native species there as well. So it was really quite an interesting thing, and everyone collected things that interested them. We collected also by the pond, where I'll show you an interesting thing we found. And you can see that we weren't particularly dressed for field work, but it worked out anyway. And one of the samples I'll show you came from this scraggly looking tree, which is actually a sort of wood that's passed off as mahogany. And it has these beautiful seed pods, which the sister is holding in her hand. And there it is. And this is a traditional African plant that is used both for wood, it's a high quality wood, but also for oil from these beautiful, beautiful seeds. Another plant that we collected among the 40 or so was this one, an interesting plant. This is a plant that's very tolerant of drought, which happens in eastern Nigeria quite a bit. And it's used to mark the boundary of households, the family plots of land in Nigeria, and also to mark off important places. So it's called the boundary tree. So it's quite an interesting local plant. And around that pond, we found this dragonfly, which looked pretty nondescript and I thought would be uninteresting, but actually turned out to be pretty interesting to me. We took those samples back to the classroom lab here at the college campus, and we extracted DNA and was pretty successful at amplifying barcodes from a number of species. Now the barcode amplicons or PCR products may differ slightly in size because in fact they vary a little bit between different organisms. But this tree here, this Akparata tree, is sample number 22, and that was the DNA that I then brought back to the United States and sent off for sequencing. Well, with that, let's take a look at and set up some experiments here. If you're following me on a computer, which I suppose you are, you can open up another screen if you like and follow along with me. Do a search for DNA Subway. The first entry in a Google search will in fact be this, and you'll come to a web page that looks like this. I'm just going to expand this a bit to give you a better view here. There we go. When you come, let me log out because I was doing a little work here previously. When you come to the home page, it will look like this. You have the option of either registering and logging in with your username and password, or you can enter as a guest. Today, I'd recommend if you are not registered, just enter as a guest and you can easily do all the experiments that I'm going to do. The benefit of registering and logging in under your own name is that you can save your work from session to session. Now, as a guest, your work will be saved for about 24 hours and then will disappear. But you'll see that I have a lot of projects that I started. It's a very stable program. If you lose connectivity or actually accidentally shut it down at any point, it will certainly come back in the state that you left it. I'm going to log in because I have some things stored here I may have to call on. If you log in, and if you have made some projects, you'll be presented with your project page here of various things that you've done over time. Otherwise, you'll be presented with this home page, and the key thing is that for everything you do on DNA Subway, you have to initiate a project into which you'll load data and perform analyses. So you can click on this box from the home page, or you can click on that same box from My Projects, either one. Most likely you're here at the home page, so I'll initiate the project by clicking on the blue square here. First thing you want to do is to define the sort of project you're doing. In this case, we're doing barcoding. I actually have a plant and an animal sample, so I have to decide whether I want to click on RBCL or cytochrome oxidase 1. The only difference that you'll see is that I will be presented with different sample sequences. So I think I'm going to go with RBCL because I'd like to have available some plant sample sequences. Then in this area, you choose where you're going to load your DNA from what source. You can enter sequences here using the FASTA format. You can upload files which may have a list of a number of sequences. But for most of the experiments that I do, and for this experiment today, I'm going to import the DNA that was actually sequenced during this field trip to Nigeria. So I'm going to import trace files from the DNA Learning Center. What you see here is a pretty substantial list of classes that have done this experiment recently, and I'd always love to look around and see where these different schools and classes are. Mine is actually, and if you've done it recently, of course, you'll be near the top of this list if you sort by date. I'm going to just go and find my samples from Nigeria, which I did in January. So I probably could find them an easier way, but I'm just going to go back in time, and you'll see a number of the different universities and places that are doing this experiment. And here's my experiment, Godfrey Okoye University in Nigeria. There's two files. I have to just make sure I get the right one here. Yes, this is correct. Now, what you'll see when you enter the file is that there are two reads or two records for each sequence. It's a little difficult here. The LC2243 is the name of the primer that we used. And this is the number of the sample here. And then you'll see that for sample 1, I have a forward and a reverse read. For number 2, a forward and a reverse read, and so on. Now, I could load all of these files by selecting all. But in fact, these files are deceptively large. They're not just files of ATCs and Gs, which are small files. They also have quality data and also the actual data that came off the DNA sequencer, which you'll see is actually visual data. So they're pretty big files. If I download all of these, it would probably take me 40 seconds. So what I'm going to do instead is to just take the samples that I'm interested in. Now, I'm going to show you another thing that you might as well get used to, is that if you do any kind of sequencing experiment, you're going to have to make yourself a chart like this. And this is the chart of the experiments that I did in Nigeria. And here's the problem, is we started off with about 50 things from which we extracted DNA. They're in this list on the left. We had to give them a number to keep track of them, a sequential number. Well, only a fraction of those actually gave amplicons or PCR products. And you'll see all the ones with an X here did not yield a product. And frankly, although occasionally you can get DNA sequence when you don't see a band, for all practical purposes, it doesn't make sense to sequence any of these that failed in the PCR. So what that means is, before I send these sequences out, the DNA out for sequencing, I have to generate a new list that does not include all the failed samples. And so in fact, then here's the sequence numbers. And I actually sequenced some of these just because I was interested. So what I'm going to go through is to look for those three interesting things that I showed you: the boundary tree, the aquarata, and the dragonfly. And I know their numbers, but I'll just look them up here quickly. So the dragonfly is, I think, 28, yes, okay. So I want to take 28. I want to take the aquarata, which is 43. And I want to take the boundary tree, which should be number 14. No, sorry, number 25. There it is. So I've just selected these samples that I want to look at. Now I'll go in and just select them from here. 28. Now, if I didn't select them here, sooner or later I'd have to figure out where these things are. So I'm just figuring it out from the beginning. There's 28. Forward and reverse read. There's number 14. Forward and reverse read. Oh, that's not what I was going to do. That's a different one. 43, 25. Forward and reverse, and 28, the dragonfly. Oh, I've done that. So I've got them 25, 28, and 43. Okay, so I have the three samples that I'd like to look at, 2 plants and 1 dragonfly. Now I just go down and add these files to the project. You'll see there's a progress bar at the bottom. They're being loaded now. Now they've been uploaded into my project. I have to give it a title. I'm going to call it Friday because I don't want to get it mixed up. I can write some description of this. And then press continue, and I'll create this project. And it's been created. If you'd like to edit any of this information, you can edit it on the fly. And just save it, and it's automatically saved. It's very robust. If I were to crash right now, everything would be saved. And you see there's my new description. Okay, let's get started on the analysis of these 3 organisms from Nigeria. First, I can go in and just take a look at the sequence. And what you'll notice, if you just look at the A, T, C's, and G's, is that in the beginning of this sequence, there's normally ends. That's where the base pairing software could not determine what the nucleotide was. You'll also see that at the end of the sequence, a sequence of ends that cannot be determined. I've pulled a wrong sequence here because this is a very bad sequence, so somehow I got the wrong number, but that's okay. We'll just disregard that one. Now, let's take a look at the so-called trace file, or the electropherogram, that actually lets you see the quality of the sequence, base pair by base pair. This is, to my knowledge, the only true online electropherogram viewer that you can find. Most of these are actually downloadable applets that run on your local machine, but this is entirely online, so it's a wonderful little thing. Now, let's take a look here. If you come into the sequence a bit, let's just start with looking at good sequence. If you want, you can increase the amplitude of the y-axis, and what you'll see is that when you have good quality sequence in the center, you have distinct peaks, well separated one from another, and very little background here. Now if I move back to the beginning, you'll see what bad sequence looks like, which is the traces of all 4 of the nucleotides more or less superimposed. This is all background. And then as you get into the sequence, it settles down. And you'll see that if I scroll through this, this is a very good sequence until I get to the very end, and then you'll see that it crashes off again. Final thing I want to show you about this electropherogram viewer is something about the quality score. It's quite important. Quality scores range from 10 to 50. The first digit of the quality score, for example, the 1 of 10, means an exponent of to the base 10. So a quality score of 10 is 10 to the first power, or 1 sequence error per 10 nucleotides. So it means that when you're looking at this sequence here, which looks to be about a 10 score, it means it's a 1 chance in 10 that this is an incorrect call. And in fact, this looks, see how this is crammed next to the peak next to it? This probably is a bad call. A quality score of 20 means 10 to the second power, or 1 sequence error per 100. So if I come into that quality score, which is right at this horizontal blue line right here, it means that there's a 1 chance in 100 that this is an incorrect call. A quality score of 30 means 1 sequence error per 1,000. A quality score of 4 is 1 per 10,000. 5, 1 per 100,000. Now, a quality score of 20, or 1 error per 100 nucleotide calls, is the threshold for high quality sequence, and we've drawn this line here. And the point I want to make is when you come into this sequence, this is high quality sequence. This call here is a little bit lower, and you can see why that is, because there's a superimposition of a second base pair there. I could come into this and edit this if I like, but usually it's not necessary. And in fact, this is very high quality sequence. Let's look at the low quality sequence here. And what you'll see, I obviously got the wrong one because the dragonfly DNA is actually fine, but I took the wrong one. This is low quality sequence, and you can see you can't make any sense out of it at all. Same with this read here, same thing. But here's back to the good reads on this sample, and so on. So that's the electropherogram or trace file viewer. Next thing we can do is just trim off those ends at the end of the sequence, and you'll see I've gotten rid of most of them. And you'll see I obliterated this whole sequence because there's really nothing to trim there. So that's the sequence trimmer. Next thing we want to do is to take the forward and the reverse read, and place them, pair them together. And then in the next step, what we'll do is the reverse complement of one of these sequences. So in fact, they're both being read as if they were in the same direction or on the same strand. So first, let's build the pairs. Pretty much you can do this automatically. And there you'll see that I've paired a forward and a reverse read for number 25, and a forward and a reverse read for number 43. If you need to, you can edit this by hand, but normally that's not necessary. So let's save those pairs. And the final thing we can do is come into something called the consensus builder, and this will show us how those have aligned. And what you'll see, just click on this to come into it, is that you gain some extra sequence here from the reverse read, where the forward read is missing sequence. Where there's a low quality base pair, these faint colored ones, you'll see that the second read can confirm that that was actually a pretty okay read. And as you come into the good quality sequence, you'll see that the forward and the reverse read entirely confirm one another. And this is the sign of very high quality sequence. You can really go to the bank with this. And at the end, you'll see that the forward read is providing some extra information. You may or may not want to use this information. Frankly, you usually trim it off. And there is a trimmer here. I'm not going to bother to do it because I rarely do it, but you can come into this and trim off, for example, that part of the sequence if you want to just get back. So I'm not bothered to do that, but if you're going to publish this, that we actually come in and trim these rather carefully. Okay, next step we want to do is to add some more sequences to our analysis, because really at this point we only have 2 sequences to work with. First thing that we can do is to do a blast search with our sequences. Now, in fact, here are our two good sequences. I'm going to blast this one first. Now, that result came back very fast. I've done this particular analysis a few times, and what DNA Subway does is to cache any blast search that's been done with a particular sequence and saves it on the server. It's a really nice thing. I didn't do it to fool you. Normally this would take probably about 20 seconds to run this blast search. But I wanted to let you know that if you've done a search before, or if anybody's done a search with this, or anybody coming back to share this analysis with me, would have this cached analysis, which is really quite nice to have. Well, this is the analysis of Akparata, that tree with the beautiful seeds. And what you'll see here is two bits of information to really pay attention to. The bits, first off, you have the number of mismatches, which is how many mismatches were there in the internal part of this sequence, not the ends, because at some point there'll be mismatches. The bit score is a combination of the length of the alignment. In other words, how long do you go before you have a lot of mismatches, that basically two mismatches in a row, and how many mismatches are in that alignment length. So here we've sort of taken all the scores, the top 20 scores from the blast search, either by the score and or the number of mismatches. So what's interesting is if I come down the list, here's some things that only have one mismatch. That is slightly lower score because they're over 576 nucleotides versus almost 600. So this changes the score just because of the length of the alignment. Now, I can look here, and if I didn't know much about Akparata, I would, as I found out when I first did this, that you can see that it's related to a tamarind tree, and if I follow this link to Wikipedia, I get an idea, well, look at that. A tamarind also has a pod. And then it comes to me that in fact, they're all in the legume family. But this interesting one down here is in fact really interesting because when I click on this, I see a seed pod that's very close in appearance to the one I saw in Nigeria. And as a matter of fact, this sequence only came into the database in the last month or so because I actually hadn't seen this one before I tried this analysis yesterday. So something always new showing up. And here's another one that's very closely related. Let's take a look at it. There's the flower. We don't see the seed pod, but my guess is it has a pod as well. So what I'd like to do is take a selection of these plants and add them to my analysis. I'll only take one tamarind. There's a number there. I'll take this thing, whatever it is. I'll take this plant. Of course, I could research any of these plants that I don't know much about, which is most of them. And I want to take this one that's most closely related, the Aphelia bella. And I think that's enough. Of course, you can take as many or as few as you like. Now I'll just add these blast hits to my project. Now while I'm at it, I'm going to blast with the other plant. That's the boundary tree, which should be this one. Now we're getting a real blast search because this one wasn't cached because I hadn't done one in a long time, so it cleared the cache. So this is the amount of time you can expect to do a real blast search. This is calling the blast server at the IPlant Collaborative, which is essentially a mirror of the blast search at the National Center for Biotechnology Information, or NCBI. But you can see it came back pretty fast. And again, I do the same thing, which is look at both the bit score and the alignment length and the mismatches. And here it's lined them up a little better for me. This seems to be the closest match here. And let's take a look at it and see what sort of tree that might be. Well, I'm just getting this record here rather than an actual picture of it. Here's another one that's relatively closely related. Let's see what it looks like. Doesn't help me much, so I'm going to have to look around. But here's something that I recognize, some relatively closely related things that are in the oleander or olive family. So I'm not that great on plant taxonomy, but normally you can sort of figure something out. Here's one that we know, Forsythia. So, you know, this is a plant that has some relationship to some of these things. This is that sweet-smelling thing, lilac, syringa. And this, so I usually take, this is actually an interesting one here. I can't quite remember what it is. I think it's a redwood. No, I guess not. I forgot what that one's called. But I'm going to take that one. And again, what I've done is just take a selection of these plants and added them to my analysis. They're all there now. Great. So we're moving along. We'll just be a couple minutes beyond 5 because we lost that disconnect time. Now, if I want to, I could add other data to this. For example, if I was sitting here with an analysis I had done previously, let's say on other relatives of this tree, I could upload these either as a whole file itself, or I could paste in a FASTA sequence. And a FASTA sequence really looks like this. It has a caret, and then a name for the sequence, and then the sequence of A's, T's, and G's entered on the next line. So that's all that it means by FASTA. It's very easy. I don't know if I can get rid of that, but let's just erase it. You can also import sequences that you find from GenBank by using the accession number, and that's very easy, or from BOLD, the Barcode of Life database, or you can import more trace files from the DNA Learning Center, add to that experiment. I'm not going to do any of that. But I am going to add some reference data, which is the eudicot plants. Or I'll tell you what, maybe I'll just add common plants. This is a little shorter thing, and we know these plants a little better. So that's some reference data that we have stored there for you. Now I've done a blast search to add to my analysis. I could upload data if I like. I added reference data. Now all I need to do is select the data that I want to do in my first analysis. And let's first do the Akparata, which is number 43. Those are the blast hits here. And let's take the reference data. Save those selections. And now let's do a multiple sequence alignment. This is using the algorithm called MUSCLE. It's related to the algorithm you may have used, which is highly similar, which is called CLUSTAL-W. You can see now it's ready to view, and the alignment is back. Let's take a look at it. And now we see something that looks like a barcode. It's pretty differentiated. There's a lot of bars here because we've compared a lot of species. So the greater the number of organisms that you do in the analysis, the more differentiated you'll see the barcode. However, what's nice about it is when you come down to this group that shook out of the blast search, you can see that they're all related by a similar barcode. And this is the nice thing about generating barcodes for a bunch of plants or animals at the same time. Now, what you'll see if I focus in is here's my plant, the Akparata. Here's the Afzelia bella. And if we zoom in a bit using this feature up here, and I'm going to focus, maybe I'll just sort of course I could just do these two on their own. But here's the two I'm looking at, this line here and here. And what you'll see is here's a sequence polymorphism that's different between my sample and the Afzelia bella. And I believe that that's the only difference between these two samples. Here they are. So I see one sequence polymorphism between my sample from Nigeria and this Afzelia bella. Let's build a tree quickly. In order to build a tree, we have to trim out the parts of the alignment. I'm just going to back out here. Well, also remember you can expand this window like this. I backed out of the alignment. I'm going to back out just a little bit more so I can see it all. Well, that's most of it. And what you'll see, these gray areas here are regions where there's no data on at least one of these samples. So in other words, this barcode is longer than all the rest, so these are just marked as gray areas. The light gray areas are regions where all of these species have the same sequence through these regions, and of course the nucleotide polymorphisms are where they differ. To build a tree, you've got to trim out these blank ends, these unmatched, unpaired ends at the beginning and the end. And we have another trimmer that's going to do that job. So notice you trim at different times. Sometimes you have to trim more because occasionally you'll get sequences that have gray boxes in the middle, which means something's not aligning and you have to throw that out. So let me expand this a bit. Now we have pretty pure barcodes here. Again, here's shaking out is our group. Here's the polymorphism difference between my sample and the GenBank record. Let's build a tree. Now, I'm not an expert at a tree, so if you ask me a lot of questions about it, I'm sure to give you the wrong answer. But let's start by building the simplest kind of tree, which is called neighborjoining, which builds the tree one link at a time, and each link will be on a set on a separate step. It's very fast to build, but it's only building, remember, out of all these data points, there are hundreds of different trees that you could build to explain the relationships between those different mutations. But here's one, and mainly I just want to come in and see that group that we generated, and yes, they've shook out there. And you'll see that at least of the things we have here, the begonia is the closest, most closely related of the common plants here. Of course, these are monocots over here, so we can understand that they're not closely related. Now I can build a second tree using another method, which is the multiple likelihood method, which actually builds multiple trees, compares them, and then presents you with one of the most likely constructions. The thing about this is you don't have each thing on a separate step, and where things are very closely related, in other words, have very few sequence differences, you'll see that it's flat, very flat here with small extensions. So these are things, again, from the BLAST search with very few sequence differences. Here's just that one difference between my plant and this Ephelia bella. Okay, and this can sometimes help you understand the relationships between things. Will warn you that take any tree with a grain of salt. You really have to pick the right set of data. They have to be carefully trimmed. And they can mainly help you to understand where you already have some knowledge. If you just use a tree blindly, I don't think it will help you very much. Now, let's quickly analyze the second tree. So what I'll do is just unselect that, select this pair. I need the blast hits that I got from that search with number 25. And let's just, well, we can leave these here. So let's get rid of them. And let's save those selections. Now I've got a new selection, a new multiple sequence alignment. And again, I'm looking for my sample here, and there it is. And what you'll see is that, let's zoom in a bit. This is to the base pair level, which is sort of overkill. Well, I've lost that screen here somewhere. Sorry, I just swiped my screen and can't find it again. Okay, well, but here's the analysis again. What I want to do is just look at these two samples here. Here's my thing and its closest matches around it. And what you'll see is that my sequence differs by one. I'm looking at this one, Comaranthus minor. 1, 2, 3, 4, 5, 6, 7, 8. So about 8 differences between my sample and this Comaranthus minor, and between the one below it, Hydranthus salaki, then you'll see that there's 1, 2, 3, 4 differences, 5 differences. So the point that I'm showing you here, last time I got a very, very close match, just one nucleotide difference between my sample, the Akparata, and the Ephelia. In this case, I'm off by a bit more. And if I trim this alignment, I can also quickly build a tree. And there's the trimmed sequence. Again, you can, can zoom in as much as you like and scroll through it to see the differences between the samples. I'll build a quick tree. And there's my sample shaking out here. And it's actually put one of the close matches on another branch. Now let's try, which is a bit surprising. So let's try the maximum likelihood. I just want to see if all of those close matches shake out together, which I'm expecting that they will. And there's the closest match here. And the other match is somewhere else. So it's showing that the affinity between these two sequences might not be as simple as you just judge by your eye. Now, this is the beginning of a real quest for knowledge, and I didn't load the dragonfly sequence, so I'll tell you the summary of this. But a lot of what you then do is what you would do normally when you're not sure about what you have, which is to do Google searches. So for example, I knew that original plant was called Akparata. That's all I knew when I came back. And when I did a search for this in Google, you see a lot of translations and they don't seem to make sense. So maybe I'll just say Akparata plant. And I haven't done this in a while. So here it is, plant and harvest Akparata seeds. Potentials of Azealia africana vegetable oil and biodiesel production. And this is where I learned that, in fact, this is sort of one of the native plants that's been looked at from time to time as a source for oil in Africa, because it grows there quite well. And the other thing is, of course, you can just go in and look at the images, and there are those seeds. And so the point of it is, from knowing nothing about this plant other than its Igbo name, the native language name, I started exploring about it, finding out different things. I also found out that it's a source, it's a very good source for the sort of mahogany that they use on outdoor decks, because it doesn't absorb water. It was a whole quest. But the interesting thing is that I looked and looked, I refined my searches, I did some other blast searches, refining the sequence. I went outside of DNA Subway and just took that Akparata sequence and took it to NCBI. I did word searches, and what I found out is that this Akparata plant is called Azealia africana. And I'll just come back in now and show you what I found out. And I'll show you a couple other things I found out. So that's the scientific name. And remember, the one that I called up was called Azealia bella. And it turns out that they're very closely related. They have slightly different range in Africa. But Akparata or Azealia africana doesn't exist in GenBank at this moment. And so I'll publish that sequence there. I'm still meaning to get around for a couple of months. Now, this was the dragonfly. And when I did a search with that, I found something pretty interesting, which was, of course, it's a dragonfly, and it was very close. And what I found was that it had a very close match called Acosoma panoportes. But you'll see it's not a perfect match, and if you actually go through and count the differences between the dragonfly that I found in Nigeria and this close match in the database, there's about 12 sequence differences, which seemed like a lot to me. So then I said, here's my dragonfly, and I went searching around for it on the internet. I didn't have a dragonfly specialist around. So I started looking around. And what I noticed was I found this one, and this is Acosoma panoportes. So I have a sequence that points that this thing seems to be Acosoma panoportes. And I have this image here, and I'm trying to say, does this look the same? And the first thing you say is, wow, the color doesn't look different, but this is this characteristic compressed abdomen here, which actually most most dragonflies don't have. And I saw this pattern that actually looks pretty similar on its back. I did some more work. This was hours and hours when I should have been writing grants. I was fooling around with this dragonfly, and you'll do it too, and your students will do it too. And it turns out that this is actually a drab female. So I learned that dragonflies have sexual dimorphism just like birds, and just like birds, the males are better looking than the females. So that was new to me, and I thought it was quite interesting. Well, then I found this out, that This species is found throughout Africa. Here's Ghana. It's found throughout Asia, India, Thailand, and so forth, the exact same species. So then I went into the database and did some more searches, and what I found was that this is a dragonfly from India, and this is a dragonfly from Japan. Same species, but a lot of sequence difference. And then if you build a tree, you can see they shake out together. Here's my dragonfly, and here's the ones from different parts of Asia. And so then it leaves me with this question. And then I looked further in GenBank. I did word searches. And what I found out is that in all of GenBank, for this genus, Acosoma, there's one other species in its genus. And for this whole genus, which contains 2 dragonflies, there's a total of about 10 sequences of any kind in GenBank. Some of them are cytochrome oxidase 1, and there's a few other sequences. So the point of it is, very little is known genetically about this genus of dragonflies. And they range throughout the world. And my question is this, and it's probably the question you have, which is, are those dragonflies really the same species? And I'm dying now. I've just had some more of these dragonflies collected for me in Nigeria because I didn't bring one home. I've got to take them to the dragonfly guy or girl, and they've got to tell me, one, is this the dragonfly I think it is this Acosoma? I'm pretty certain it is. And secondly, where are the other dragonfly people, and are they studying the genetic diversity of these dragonflies? Because in fact what we're finding is a lot of times when people thought the same organism was the same across a big range, sometimes there's what's called a cryptic species there, another species that's sitting there, and you can differentiate them, sometimes by behavior or other things, but you can differentiate them by sequence. So I'm left with this conundrum. I don't know anything about dragonflies. I learned a bit. It's very interesting. Also, dragonflies migrate thousands of miles like birds, some species. So that's probably how this one got around. So, and there's another tree that I built. Oh, and here's the different records. Oh, and this focuses in on these. And look at the number of polymorphisms between these different pairs. It's really quite a lot. More than you generally would expect in these systems, although this is also showing you that in some taxa, you get a lot of difference at this sequence position, cytochrome oxidase subunit 1, this barcode region, and in some you get very few. So this is what I found. All these new sequence polymorphisms in this drab dragonfly flying around a pond in Nigeria. And what I'm trying to say to you there is the same thing will happen out in the pond in your backyard, because we went down to the Cold Spring Harbor, had kids working there for a couple of summers. They find a pretty nondescript looking fly, and it turns out that fly is not in GenBank. They find a pretty nondescript looking butterfly. We can find out what genus it may be in, but it's not in GenBank either. So the point of it is, there's so much diversity in the world around us. We only know a fraction of that. And you can just go out, do some DNA barcodes, and like me, try to find out something about something you know nothing. And here's what I found about this boundary tree. I could show you the analysis. Very interesting local tree, important to their society for making boundaries, setting off monuments and so forth. And what did I find with this? Here's the sequence analysis. And what did I find? Here's my tree here. It's not in GenBank either. And it's related to lianas and jacarandas. This was a sequence that we actually published, and my sequence came up in the database search. So here's another new sequence record, and it's a lot of fun. So that's really all I wanted to show you. There's lots more I could show you. The main thing I have to say to you is, with yourself, be creative, follow the leads, find out something new, and just encourage your students to do the same thing. The dragonflies, it's not that big a deal, but it was really interesting to me, and I learned a lot, and I think you and your students will also learn a lot, regardless of what you do when you try DNA barcoding. So I'll stop now. I'm a little bit late, but I'll take any questions that you have. If you have a question, you could just type it into the chat area there. I'm sure it was perfectly clear, everything I went through. A few people are typing, so I hope we have some questions here. I'll read them off as I get them. Well, seem to have lost those. Can we open up the audio side? We're still learning how to use the system, so I'm not sure we can open up your audio.
If anyone wants to raise their hand, I can try.
Also, if you raise your hand, we could try to call on you.
It's saying multiple people.
Here come some things. If I used 4 primers, so here's the question. If I used 4 primers, 2 forwards and 2 reverse, can I use DNA Subway to pair them? Well, let's just assume that your primers are focusing in on the same region, because in fact, many times we use degenerate primers. We use a mixture. So long as those primers are amplifying the same region, you'll generate a barcode and you can analyze it nicely. Now, if they're barcodes for 2 entirely different regions, you're going to have a mess, because you're going to have superimposed trace files. But yes, in fact, in DNA barcoding, it's sort of rare that you can use one primer, exactly one primer forward and exactly one reverse primer, and amplify a range of species. Typically, there is actually a mixture of primers there anyway. Did that answer your question? I hope so. Here's another question. Are there many algae in the databases for barcoding? I haven't looked. I only know, a small set of things that I've sort of played around with. And of course, I'm not an expert in barcoding. But here's what I can tell you. For things that are small, out of the way, and non-charismatic, meaning not great to look at, there's a lot of things that are not in the barcode in GenBank. So, for example, if you look at things, let's say, several millimeters or smaller, of which you'd of course have many algae that would fall into that, I can guarantee you that you will find new GenBank records. Let's see. Here's another question. I was wondering what other types of alignment applications, such as T-Coffee, Clustal Omega, and Clustal W2, will be available at DNA Subway. We don't have any plans to build additional aligners into DNA Subway. I suppose if you talked to me or emailed me and gave me a compelling reason why we should do that in education, then we probably could. Generally, we try to make DNA Subway have a high level of utility, but not do everything. So what I'd recommend is that if you really wanted to train students in using lots of different alignment tools, start with DNA Subway to get them limber and used to things, and then turn them loose on those other tools. And we find that this actually works very well. Our reports from the classrooms is that using DNA Subway to help kids get an idea of how databases work, how you do searches, how you gather sequences together is very good, and that the things they learn in DNA Subway seem to be transferable to other databases and other utilities. So that would be my answer about using other alignment programs. There's one more question seems to be coming through. Okay. I'm not sure about algae, but about 350 of 1,600 lamb plants in Delaware lack barcoding. Great, where'd you get that statistic? So there you go. Here's the point, is that you may think that in an urban area or something that everything is known. But for example, we had students in New York City who barcoded fungi in Central Park. And that was the first time that that kind of study had ever been done in Central Park before. So, people tend to, here's what I learned, which I think is telling. If you try to find a range map of species that's actually based upon observations, so for example, let's take a map of the United States and try to determine the range of some species, not extrapolated from a few samples, but based on a lot of data. Do you know that the only range data that we have in the United States are for large species, charismatic species, vertebrate species, especially mammals, but the biggest database we have is on birds because they're fun to watch, they're charismatic, and a lot of people do it as a hobby. But everything else falls short. So even if we may have some data about this algae being present here, we don't necessarily know if it's present in Delaware. So we need a lot, if we want to understand species distribution and ecology, having lots of people doing barcode, sending in those, putting those samples in the database, and having spatial geographic information so we know where they came from is really very important, because we can't even build range maps for anything. We couldn't build a range map of your average lizard around the United States, much less some copepod or small thing. It's a fascinating field, even if you're a non-expert. A lot of us as teachers have to be generalists. But this is a great thing. It's fun. You can explore it with your students. I learn something every day. As soon as I start doing the barcode and stuff, I don't get any work done. So I'll warn you about that as well. So we don't seem to have any more questions. It's been great being with you for this short period of time on a Friday afternoon. I hope you have a great weekend. I hope I see you again either in cyberspace or the real world. So good afternoon to you.